In this video, we will be going over how mRNA is translated into proteins. So we're going to find out how proteins are made. Now in the previous video, I said that RNA polymerase reads from the 3' to 5' and it lays down nucleotides in the 5' to 3' direction. Now a nucleotide is one of these letters. So C stands for cytosine, that is a nucleotide. U stands for uracil, that is a nucleotide. And of course, C is another nucleotide. Whenever you get 3 nucleotides together, they create a codon. So typically, 3 nucleotides gives us a codon. And a codon is just a chunk of information. So this is a chunk of information. Right there. Okay. And the way proteins are made is that a tRNA, so a tRNA, which we also discussed in the last video, will read these groups of information, these chunks of information, and they will create amino acids. And these amino acids are the building block for proteins. So if tRNA were to read this, it would read it. Let's just make up a tRNA molecule. So let's say I start reading. And when it reads from the 5' to the 3'. So here's the 5' end, and here's the 3' end right there. And when it reads UCA. And UCA actually codes for serine. So we go to the U block, and we go to UCA, and that is serine. So again, serine is an amino acid. So when tRNA reads all these information blocks, right, it creates a protein. So serine, alanine, leucine, proline, those amino acids create a new protein. And let's say that protein codes for, I don't know, a hair or something. But that's how proteins are made, and they're made by these codons. And so now we are going to investigate how this occurs, how this process takes place. But before we can do that, we have to know that there are about 61 codons. There are about 61 codons that code for amino acids. So we call that AA. And there are 3 codons that code for stop. So when tRNA reads a stop codon, for instance UGA, let's write it over here, UGA, it reads it and it says, hey, this is a stop codon. You know, I have to stop making these amino acids, and the process of making proteins stops. Now, there is a start codon, and a start codon is one of the 61 codons right there. So a start codon is actually methionine. So notice that there is only one codon that reads for methionine, and this codon is called AUG. And again, this is from the 5' to the 3' direction. And whenever tRNA reads AUG, it will actually code for methionine. So within eukaryotic cells, AUG codes for methionine, and methionine begins the process of creating amino acids, and thus it begins the process of creating proteins. Now, what are the stop codons? Well, the stop codons are pretty easy to remember because they all start with U. So we have U. We can also have UGA, U, A. And what else can we have? We can have U, A, G. So notice that all of the stop codons start with U. One of them have UAA. We can also have UAG. But what if we switch this guy? What if we do that? We get UGA. And so they are very easy to remember, the stop codons. And AUG is the start codon. So if you're taking an exam, then I recommend that you just memorize those 4 codons, because sometimes that will show up on an exam. They'll say this codon UAA is coded for. What will happen? You said, oh, okay, it will actually stop the process of creating proteins. And that would be the correct answer. So remember those 4 amino acids, those codons, and that AUG codes for the amino acid of methionine. Now, codons can actually code for the same amino acid. Notice that UUU, UUC both code for phenylalanine. And sometimes multiple codons code for the same amino acid. For instance, when we have, let's see here, we have leucine being UUA. We also have UUG for leucine, but this whole section right there also codes for leucine. And so we have about like, I don't know, like 1, 2, 3, 4, 5, 6, 7, like 7 different codons coding for the same thing. And so this allows us to do redundancy. So the code for amino acids are very redundant. And that kind of allows us to do like a failsafe. What if I don't have any UUAs and I need leucine? Well, I can have a UUG and I'm safe. I can still produce leucine as long as I have one or the other. Now, if I don't have either of those two, I have 5 more codons that can code for leucine. And so there will almost be no way for me to not have leucine because I always have codons. And that's kind of like a fail-safe mechanism that organisms use to create proteins. Now, there's something that is almost always tested on, and that is called a wobble. So wobbles they sound very cute, but what are wobbles? Well, wobbles are kind of like changes in the third nucleotide of a codon. So, for instance, U, U, G, and U, U, C, that is a wobble. Why? Well, this is the first nucleotide, that is the second nucleotide, and this is the third nucleotide. Correct? 1 and 2, they are the same. They're not bonding, by the way. I'm just showing that they're the same, right? So these are arrows. But notice that G is different from C. Okay? Well, since we only did one change on the third nucleotide, it has to be the third nucleotide. Cannot be anything else. The protein itself isn't really going to change that much. It's pretty stable. But notice that we're not coding for a leucine anymore. Notice that we're coding for phenylalanine. So sometimes you can get away with this, right? For instance, if I had UUA and UUG, that is also a wobble, but they code for the same thing. This would be leucine, right? But sometimes if they code for different proteins, or I guess different amino acids, that can have a detrimental effect. It can be a really bad thing. So sometimes wobbles can change the reading codon, but they still code for the same amino acid. Sometimes wobbles change the amino acid completely, and that would kind of be unfavorable, right? That can destroy the whole protein or disrupt stability. So you have to be really mindful for it. Okay, so now you know what a wobble is, and now you know that it's just one modification of a nucleotide in a codon, right? So these make the same amino acid, and they have one modification. These make different amino acids, and they have one modification. And it's mandatory that it's on the third nucleotide. And now this is tRNA. And tRNA is the main, it's the main guy that starts doing the translation between mRNA and proteins. So this right here is tRNA. Now tRNA has 4 double helical regions. So if we count, that is 1 region, that is 2, 3, and 4. Okay? So those are the regions. And notice that it has 3 loops. Okay, so it has 3 loops, 1, 2, and 3. So that's kind of like the anatomy of tRNA. And the 3D structure is kind of like an L shape. So it's an L shape. For some reason that's almost always on exams. Kind of weird. Anyways, so it's an L shape. So now we have to look at the anticodon because everything else is kind of meaningless except for the anticodon. So the anticodon for this guy, as you can see here, if we go from the 5' to the 3' direction, the anticodon would be this guy, right? So let's redo this 5. So 5' would be G, A, and then 3' at this end. But the most important thing that you have to remember, and possibly the most difficult thing in this course, is knowing how to read anticodons. So yes, it is from 5' to 3' GAA, but whenever the tRNA is being read and being it's in the process of creating a protein, it attaches to the mRNA from the 3' to the 5'. Kind of weird, right? What I'm saying is the tRNA is going to have its anticodon, the anticodon. So here is going to be the 3' and here is going to be the 5'. So if we go from the 3' we have A, G. So now we would have this. We would have A, G. So now it's zipping along the mRNA. So let's have our little mRNA right there. And this is from the 5' to the 3' direction. Now it's going to read the complementary strand. So what's complementary to A? An mRNA. So, since this is RNA, it is not going to have thymine. It would actually have uracil, since this is RNA. So it would have U, C. And so this protein would be created from UUC. Okay? So tRNA will take the UUC that it reads, and it will actually produce from this bulb on its head a amino acid. So the amino acid could be phenylalanine, and it can also be serine. And so what I'm saying is, within the mRNA, the UUC already existed. We didn't create it. The tRNA just has what it wants. It says, well, I need something that is complementary to the AAG. Oh, UUC is complementary to the AAG. Therefore, I should make the protein that the mRNA codes for. Sorry, not protein, but amino acid. So this amino acid that UUC codes for is phenylalanine and also serine. Okay, so sometimes, and you'll see this in textbooks, sometimes you'll see these weird symbols. For instance, what is D doing there? And what is this little candlestick looking thing doing there? Well, respectively, if we were to look at it, if we have this, and I was never really good at physics, but we have that symbol and we also have D. And what D symbol stand for is kind of like, this is a pseudo-uridine, if I remember correctly. So this is a pseudo-uridine right here. And this right here is dihydrouridine. So these are some unusual bases, but they're not really important. I guess they would be important if you're in these upper level graduate courses. Before just undergraduate biology, they're just kind of there for just to be there, right? So they're not important for us right now, and they're not important in this course, so don't worry about it, though they do look really cool. And here's another picture illustrating what I just said to you. So notice that the anticodon in the tRNA has the 3' A, C, C, and the 5' right there. Now, it's reading the mRNA, and it will actually create the amino acids from the mRNA. So the mRNA is going to be complementary, and it goes from the 5' to the 3'. So this is going to be mRNA, and this is what actually makes the amino acids. So we will have U, G, G. And so UGG actually codes for tryptophan. So tryptophan, which I believe is TRP, that's an amino acid. So hopefully you'll understand how tRNA reads and how it creates proteins, because when I was an undergraduate, this was very difficult to understand because they would ask questions, and let's see if I can make up a question. So for instance, they will ask if the anticodon has something like, let's see, AAC, and this is the anticodon, what will we create? And so some people, they would say, oh, that's easy. We go into 3 to 5, and this is going to be mRNA. And this is wrong, by the way. This is wrong. So they say, okay, well, this is C. No, they'll say this is U, G, and they'll look at their little table, and they'll say, oh, it's this amino acid. But that's wrong. Do you know why it's wrong? Because the first thing they did was that they read the anticodon in the incorrect direction. So they read it from the 5' to 3', but that's wrong. Don't ever do that. You have to read the anticodon from the 3' to 5'. So let's do that really quickly. So I would convert this into this right here. So this is going to be the correct answer. So over here we have 5'. I'm going to flip this. Okay, so we would actually have 3', C, A, and that would be the 5'. And this is the tRNA. Now, if I wanted to code for this, I would have the complementary strand of mRNA. So the mRNA would go in the 5' to the 3'. And this is going to be mRNA. And we would have G, U, U. And so that would be the correct answer. And by the way, this would actually code for the amino acid valine. So never ever read the anticodon from 5' to 3'. That's wrong. Don't do that. Instead, read the anticodon from 3' to 5'. Right? So you can think of anti as antagonist. So it's the opposite of what you would expect. So you would expect to read it from the 5' to 3', but really you read it from the 3' to 5', and then you make the complementary strand of mRNA. And the mRNA is what you code for valine. So this would not be a good thing to code, right? So this is not the correct answer. You would say, oh, we code for this amino acid. No. You don't make amino acids using the antisense strand. Instead, you make amino acids from the mRNA strand. Hopefully that clears it up for you. Now, within organisms, we have ribosomes. But what are these ribosomes made of? These ribosomes are made from rRNA. Now, there is a difference between eukaryotic ribosomes and prokaryotic ribosomes. And the difference is the size of the ribosomes. So eukaryotes, they have the 80S ribosome. And the 80S is kind of like the overall size of the ribosome. So this would be the 80S ribosome, and it's comprised from two parts. It has a large subunit and a small subunit. Now, the large subunit has about 60 parts within the subunit. So we call it the 60S because it's after some scientists, and scientists are very egotistical. If they discover something, they say, oh, you gotta put my initial behind it. Who cares, honestly? Anyways, so the large subunit for eukaryotes is 60, right? And the small subunit is going to be 40S. Well, you're saying, well, how did they add up to 80S? Well, I know it's not the correct way to think about it, but this is the way I do it. So 60 minus 40 is 20. Okay, 60 plus 20 gives you 80. That's it. And so you have the 80S ribosome. And so what you should remember is that eukaryotes, they have the 80S ribosome for the big unit, and then they have the 60S for the large unit, and then they also have the 40S for the small. And so that will be on exams. They'll say, what is the difference between prokaryotes? And they'll be like, true or false. Eukaryotes have the 50S ribosome, and you'll say, no, that's incorrect. They actually have the 60S ribosome. So that's very important, and that you have to remember it, okay? For prokaryotes, the large subunit will have to be 70S, okay? Or I guess the overall subunit, okay? Now the large subunit will be 50S, and the small subunit would be 30S. And again, 50 minus 30 is 20, ergo 50 plus 20 gives you 70. Okay, so that's just kind of how I remember it, and let's write it over here. So prokaryotes, they have the 70S overall, and they have the 50S for the large subunit, and the 30S for the small subunit. In general, for eukaryotic organisms and prokaryotic organisms, the large subunit is the one that actually catalyzes the peptide bond formation. So this one catalyzes peptide bond formation. So we have peptide bond formation. The large, so that's the large subunit, and then the small subunit is the guy that it kind of matches the tRNAs. So again, this one matches the tRNA to codons. Right, so it's going to be facilitating the connections between tRNA and mRNA. Right. So let's look at how this occurs in detail. So here we have a very nice looking ribosome, and this ribosome actually have one binding site for mRNA and 3 for tRNA. So here is the one binding site for mRNA, but over here you have 3 binding sites for tRNA. So tRNA is going to be read from the 3' to the 5', and the mRNA again is from the 5' to the 3'. Right? So now you have the A site, the P site, and the E site. So the A site actually stands for amino acyl. That's an L, so let's write acyl, and this is tRNA. And the P, it stands for peptidyl tRNA. So peptidyl is the one that actually catalyzes the formation of peptide bonds. So when the amino acid is being transferred into the P site, it's going to form a covalent bond. So pretty interesting. And the E site is actually the exit. So this is called the ejector site. Okay, so that's kind of like the main parts of the ribosome. Now within eukaryotic organisms, there's about 6 to 9 amino acids processed per second. So we can say that we make 9 amino acids per second. Now for prokaryotes, it's pretty surprising, but they can actually make 21 amino acids per second. So they're a lot faster than eukaryotes, which is, again, pretty interesting. And here we have the process taking place. So there are about 4 steps to make an amino acid. So here we have the ribosome in its entirety. So we have the large ribosome subunit, and we also have the small ribosome subunit. Here, the small ribosome subunit has bound itself to the mRNA. Now notice that we have these little things right there. These are the tRNAs. And remember that tRNAs, they have the anticodon. So again, the anticodon is read from the 3' to the 5'. That's how it's read. Now in step 1, the tRNAs are being captured by the large ribosomal subunit. So as you can see, we are already in the process of creating this amino acid sequence. Now notice that this amino acid sequence jumped from the 3 codon to the 4th codon. Okay, so now whenever these amino acids are growing, they're going to be jumping into the newly added tRNA. So 4 just entered the subunit, and it will inherit the growing amino acid chain from the previously entered tRNA. Okay, so 3 entered first, but then it's going to transfer its sequence into the newly entered tRNA. Eventually, the ribosomal subunit is going to shift towards the right. And when it shifts towards the right, the 3 codon, in this case, it will enter the E site. And when the 3 codon enters the, not the codon, but the tRNA, when it enters the E site, it's going to become ejected. Now notice that when it ejects, the 4th tRNA enters the P site. So now when the 4th tRNA enters the P site, it will actually transfer its amino acid sequence into the newly entered tRNA. So this cycle will repeat. So let's do this again, right? So first of all, a new tRNA enters the large ribosomal subunit. Therefore, the previously entered tRNA is going to transfer its sequence into the new tRNA. When this happens, the large ribosomal subunit will shift towards the right. And when this occurs, the 3rd codon, or tRNA in this case, is going to enter the E site. And when that happens, it becomes ejected. And when that occurs, the newly entered tRNA is going to be in the P site and is going to transfer its amino acid sequence to the newly entered tRNA. And so we can say that whenever an amino acid is being created, or the sequence, it's created from the 5' to the 3'. And also, the N-terminal is made first. And this makes sense because we go from the N-terminal to the C-terminal. Pretty cool. And so this kind of reminds me of some questions that I had in exams and school, and sometimes people would just randomly ask me stuff, you know, it's pretty cool. So one of the questions would be, let's say that we have 3,000 nucleotides. I remember that a nucleotide is like A, G, C, U, etc. So let's say that we had 3,000 nucleotides. How many amino acids could we make? And you're probably wondering, well, how does that make sense? Well, notice that an anticodon requires 3 nucleotides. Therefore, since we have 3,000 nucleotides, if we divide them by 3, we can get what? We can get 1,000 amino acids. And so you'll often see that on exams and stuff, right? So if you had 1,500 nucleotides, you can only make 300, you can only make 500 amino acids. Let's say that I had a modification. So I have AUC, and then something was modified, like a mutation occurred. And so now I inserted something else, AUCG. How will that affect the amino acid sequence, or how will that affect the protein formation. And so one of the possible answers could be that you shift the reading frame. So instead of reading AUC, you now read UCG, and that could code for a different amino acid, maybe. But another answer choice could be that the C terminus will become affected. And that would actually be one of the correct answer choice. The other one would be a possible reading frame shift. But the reason why we actually do a modification for the C terminus is because we go from the N terminus to the C terminus. And so if I do a point addition, which is what this is called, right? We added a nucleotide. This can actually change the C side. So if you think of that like this, N side to the C side, adding something to the reading codon can actually modify the C terminus. So that was never taught to me in undergrad, but I had to learn that to myself, right? So now I'm sharing that knowledge with you. So before we can even begin making proteins, we have to make like an initiation. So right here we have a translation initiation factor, and this protein or this factor gets an initiator tRNA. Now remember that methionine actually codes for AUG, right? And so whenever AUG is read, methionine, which is already present, will begin to expand. It will begin to create new amino acids. So this translation initiation factor has the small ribosomal subunit and the initiator tRNA. Now it begins reading the mRNA from the 5' to the 3'. And when it reaches the AUG mRNA codon, it leaves the initiator tRNA. And this initiator tRNA starts sending signals. And these little signals are picked up by the large ribosomal subunit. So the large ribosomal subunit binds to the initiator tRNA, and now it can begin the process of creating amino acids. And so really, to get the ball rolling, you have to have the translation initiation factor, which connects to the methionine, which is always the initiator tRNA, and eventually it's going to read AUG. And when it reads AUG, it sends a signal, it binds to the large ribosomal subunit, and it creates more amino acids. And now, a charged tRNA can bind to the A site, and when this occurs, a covalent bond is going to form, because again, methionine is it's in the P site, and that has peptidyl, and peptidyl is it serves as a catalysis for peptide bond formation. So again, peptidyl serves as a catalyst for peptide bond formation, and that's in the P site. So again, methionine is in the P site. We have the acceptor site open. A tRNA is going to be accepted into the A site. The methionine in the P site is going to use peptidyl to create a peptide bond, and that is connected. Now eventually this large ribosomal subunit will move to the right, and when it moves to the right, the methionine is going to enter the E site, and therefore it is ejected, and the process will continue. But eventually, all good things must come to an end. And so eventually we will reach a stop codon. So the stop codon, again, would be UAG. We have another U, we can have AA, and again, we can have UGA. So again, these are stop codons. When the tRNA reaches a stop codon, it actually doesn't recognize it. It doesn't recognize the stop codons. So we can say that tRNA does not read not tRNA, but we could say stop codon does not read stops, right? So what actually happens is that a binding release factor will recognize the stop codon. So here is going to bind to the A site, the binding factor or binding release, right? And so when the release factor binds to the A site of the tRNA, it becomes incorporated. It becomes like it's like a little worker that clocks into work. And whenever it reads the stop codon, it facilitates the release of the amino acid sequence. And it does this by using water. And so we can say that when the release factor reads stop codons and binds to the A site and binds to the A site. And whenever that happens and it wants to release the amino acid sequence, so it wants to stop it, it uses water. So it uses water to cut peptide bond. Okay, so it uses hydrolysis. Right there. Okay, so that is kind of like a hydration reaction or hydrolysis. So to recap, in order to stop this process of creating amino acid sequences, which create proteins, we need a release factor. And this release factor goes into the A site of the ribosomal subunit. And when it does, it will actually use water to cut down this amino acid sequence. And when that happens, the whole ribosome explodes. It doesn't really explode, it just dissociates. I'm being dramatic. But it dissociates, and now we don't have any more protein synthesis until we need it. And the reason why we use a release factor is because tRNA does not read stop codons. It actually uses the release factor. And so within an organism, you do not just have one ribosome doing all the creation of proteins. That would be really inefficient, right? You have a million, trillion mRNAs in your body, and so you need more than one ribosome to do this. And so within one mRNA, you can have multiple ribosomes in different sections coding for amino acids. So we can have amino acids over there, amino acid right there, etc. And so eventually they will actually create amino acids at the same time. And right here we have a nice picture where we have these ribosomal subunits creating amino acids. And it's very beautiful. It's kind of weird if you think about it, but I think it's beautiful. Anyways, what I'm saying is this can occur simultaneously. So we can have multiple subunits creating proteins at the same time. You're probably wondering, whenever we make the proteins and we don't need it anymore, how do we get rid of them? And to that I say that we use proteasomes. So proteasomes, these are little guys that we use to break down proteins. So a proteasome contains a cylinder, and that cylinder is made from proteases, which are enzymes. Now these proteases, they form active sites and they face the inner chamber. So here we have a protease. Let's write that better. Protease. So this right here is a proteasome, and this center is a protease. Okay? Now this protease has active sites all along its face inside the center cylinder. Now when a protein that is no longer needed enters this active site, it begins to get cut. So this protease cleaves the peptide. And when it's cleaved, it becomes inactivated and it begins to be degraded. So now this protein is degraded by protease. Okay? So that is how we can get rid of proteins. So again, to recap, proteasomes break down proteins by using protease, and protease can be found within the center cylinder of the proteasomes. You're probably wondering, well, how do we know when to break down peptides? If we don't know when to break down peptides, we're going to just break down all of the peptides in our body and then we would die. But our body is pretty smart, surprisingly. Instead, what we have here is that we are going to be using some ubiquitin. So we have ubiquitin. And ubiquitin in general is kind of like a signal marker. And it marks things that need to be degraded. So, we can write this. It says signal marker, and we can say makes things break down. So, for instance, it's kind of like a message. So, whenever something is marked with polyubiquitin, the proteasome is going to open up, and when it opens up, it accepts the peptide, right? So the protein cannot enter this site without having a ticket, and this ticket is the signal of polyubiquitin. So whenever it presents the ticket to the proteasome, it says, okay, I see that you need to be degraded, and so I'm going to take your ticket, I'm going to throw it away, which is what happens here, and then I'm going to accept you into the active site. So now whenever the protein, which has the polyubiquitin, enters the proteasome, it enters the protease. Right? So it enters the protease, which is the cylinder, and it begins to unfold. So remember that peptides or proteins are folded in their active site, right? So, sorry, not the active site, but the active form. So when proteins are active, they're folded. But what happens here is that protease will deactivate the protein by unfolding it. And whenever it unfolds, it has more exposure to the active sites of the protease. And when that happens, it begins to get cut and it gets cleaved even more. And so eventually we have protein degradation. So to recap, when a protein needs to be destroyed, it is marked with ubiquitin. And that's for anything in the cell. You can have a lysosome that is marked with ubiquitin and it would actually be degraded. So in general, when something is marked with ubiquitin, it's going to be destroyed. So when protein is marked with ubiquitin, it presents the ubiquitin to proteasome. Proteasome opens the, mouth of it, and it accepts the protein. When this happens, it gets rid of the ubiquitin, and it enters the protein, and it unfolds the protein. And this allows for more exposure to the active site of protease, and it becomes cut down. So, after that whole fiasco of proteins being broken down, let's watch a video about how proteins are made and how everything that I just taught you ties together.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
And that concludes the lecture over tRNA and how protein is translated. So hopefully you understand how protein is translated, how we even make mRNA, how we splice exons and introns and whatever we do. And hopefully you understand how genetic information is processed and made. So thank you so much for watching these videos, and I hope that you have a great day. And remember that I love you, and that you are really, really cool, and I'm glad to have known you. Alright, I love you, and take care. Goodbye.